We are for part 2 or 3, depending on how you want to look at it, of the renin, angiotensin, aldosterone system. Again, I say part 3 because um, EDH is also integral in this sort of group, right? You want to think of angiotensin 2, ADH, and aldosterone all sort of working together in an integrative fashion to boost blood pressure and blood volume. So again, it's the renin, angiotensin, aldosterone system, but if you wanted to add another A in there for ADH, I wouldn't have a problem with that. So this video will focus on the aldosterone aspect. Aldosterone is secreted by the zona glomerulosa of the adrenal cortex. So let's put that as our target, our target glandular tissue for the stimuli, and then we'll focus on what those stimuli are. Right, so it's the outermost zone of the adrenal cortex. As we've mentioned before in a previous video, angiotensin 2 is a stimulus at the adrenal cortex for aldosterone secretions. We can have our arrow towards aldosterone secretion rising. In addition to angiotensin, well, let's review a little bit. So our primary input to angiotensin 2 is low blood pressure, right? So the renin-secreting JG cells, juxtaglomerular cells of the afferent arteriole, are sensitive to the pressure in that vessel. So there's the afferent arterioles are baroreceptors essentially. And when pressure is low, renin secretion goes up. So low blood pressure there is going to stimulate the JG cells to secrete renin without all those steps in here. Angiotensin 1 to angiotensin 2 through converting enzyme. And now we've got our stimulus to the adrenal cortex. Now, in addition to that, we could also add low GFR, right, glomerular filtration rate. And yes, low blood pressure would lead to low GFR, but the low GFR itself is detected at the macula densa. Again, go back to the previous video. The macula densa sends signals to the JG cells to indicate that not enough filtration is occurring. Renin rises, angiotensin 2 rises, and you get efferent arterial constriction to solve the GFR problem. Now, in addition, you get systemic vasoconstriction, right, which increases, and so we could have our angiotensin 2 causing an increase in total peripheral resistance, right, and that we've seen before. Okay, and well, we can even add this just to have it complete picture here. Angiotensin 2 stimulates EDH. Among other things, as we saw in a separate video. ADH, of course, increasing blood volume and indirectly, therefore, increasing blood pressure. Okay, so let's follow up on other stimuli to the adrenal cortex outside of angiotensin 2. The big one is, and so kind of unrelated to the blood pressure scenario that we're talking about here, but it is the primary stimulus, is hyperkalemia, right? Hyperkalemia being an excess of potassium in the extracellular fluids. Right, and I'm going to put a plus next to that one because that is definitely the larger, more potent stimulus for aldosterone secretion. And the rationale there is that high extracellular potassium is dangerous. Dangerous because potassium is high intracellularly relative to extracellularly. So if extracellular concentration rises, it tends to oppose the electrochemical gradient of potassium to leave the cells. But is a very big deal when you talk about excitable cells that require potassium efflux for repolarization. So think neurons and cardiac muscle, skeletal muscle. Well, cardiac muscle obviously more important. So high extracellular potassium dangerous because can oppose that repolarization, leading to abnormal contractions and or abnormal neuronal function, neither of which is good. Okay, and as I've probably said before, lethal injection is essentially excess potassium, right? So bad idea to have high extracellular potassium levels. And we'll see how aldosterone solves this problem in a moment. But before we do that, let's throw in the last one here. And this was a relatively weak stimulus, and it is hyponatremia, right? So this was hyper, so high potassium. Hypo was low, and natremia for sodium. Right? And so it will help elevate, aldosterone will help elevate low extracellular sodium levels. All right, and so how does aldosterone do this? Well, aldosterone acts fairly exclusively on the distal nephron, so it's going to act on what are called the principal cells. Of the DCT and the cortical collecting duct. Okay, so we know what the DCT is, right? That's the segment, kind of short segment of the nephron after the loop of Henle. We know what the collecting duct is. The cortical collecting duct is the first part of the collecting duct that's still in the cortex. It's the part that has not yet entered the medulla. What that really means is that the cortical, and this is true, the cortical collecting duct is actually very similar to the DCT in terms of what hormones it responds to, what transporters it expresses, things like that. Those two regions are very similar. And so these principal cells, in response to aldosterone, are going to change their protein expression. Aha. Aldosterone is a steroid, right? As a steroid, it is lipophilic, meaning it will bind an intracellular receptor, and that hormone receptor complex will bind an HRE, or several HREs possibly. And those HREs, the hormone responsive elements, right, are tied to, they are part of the promoter region, and they are tied to altered expression of components of the sodium-potassium pump, as well as certain ion channels. And so what we get is an increase in expression of the sodium-potassium pump, as well as an increase in expression of a channel known as the epithelial sodium channel, or ENaC. Now, what I want to do is I want to show you how they fit into the wall of the DCT, or cortical collecting duct, to show you how that we can alter transport of the ions and solve the problems that we're talking about over here. Okay, so I'm going to draw a single cell of, focusing on a single cell of the wall of this distal nephron. That's what we have here. Exaggerate. Right. So here's your other cells, right? Without drawing the other cells, right? So this would be the lumen of the duct of the nephron, right? And so the tube is sort of coming in and out of the paper here, right? And the tubular fluid is running, in and out of the plane of the paper here, right? So this is the lumen. This is the cell, right? We can put a nucleus in here, each one of these, I guess, if you want to, right? And again, all the way around to form the whole. I guess I can complete that. Enough room, right? Right. Focusing on just this one cell, though, just to show you the whole nephron segment. And so the sodium-potassium pump is always found expressed in the basolateral membrane of the nephron cells. That's probably true of all epithelial cells, to be honest. Right, epithelial cell, this is an epithelial cell, right? Found lining open space. And so we can put the sodium-potassium pump into the wall here. I'm going to find a different color. Oh, sorry, basolateral, right, is the attached side, right? This would be against the interstitium and other tissues, right? This is the free side, so that's the apical membrane. So apical membrane, basolateral membrane. So we have basolateral expression of the sodium-potassium pump. And of course, as we know, the sodium-potassium pump moves sodium out of the cell, pumps, I should say, sodium out of the cell and pumps potassium into the cell. And that's always true, right? That's true in pretty much every cell on the planet. Now, and again, that sodium gradient, largely the sodium gradient can be utilized to do work, transport work, signaling work, right, in terms of depolarization currents and things like that. The potassium, there is a significant potassium leach channel. So the potassium does a little bit of this, right? It's kind of going around and around and around. So the combination of the two really generates a sodium pump, right? Even though it's called the sodium-potassium pump, the potassium's kind of going around and around in circles. Now, without getting into it, the potassium does, yes, it's going around, but it is in much higher concentration inside than outside for reasons we don't need to get into right now. Okay, so our aldosterone is going to, activate transcription translation in the nucleus and increase expression of the subunits of that sodium-potassium pump. So we're going to move more sodium out, and we're going to move more potassium in, right? The more sodium out, let's add our capillaries, associated capillaries here. These would be the peritubular capillaries, right? Capillaries surrounding the nephron segments. Put a red blood cell in there, so you can remember. Right, so that extra sodium reabsorption here is going to move into the capillaries and be carried away and improve our low sodium levels. Now, we have to get it out of here to keep moving it out of here, right? And so that's where the other guy comes in, the increased expression of ENaC. Well, that's apical. And so we can add that guy over on this side. Right, so we'll call that guy ENaC. Sorry, not a lot of room here for big labels, right? That's ENaC. And so what we're going to do, right, so an intracellular sodium is kept low by this pump. And so this is extracellular fluid as well, modified because it's being modified by the nephron prior to this region. But the sodium is still higher here than here. So sodium is now given an increased permeability across this apical membrane. And so sodium reabsorption is going to increase, and I can really just almost connect these lines. And now we have this enhanced full pathway across the nephron wall to drive sodium movement back into the bloodstream, right? Increased Na+. Solving this problem. Now, because sodium is a major extracellular ion and because water follows enhanced solute movement, we're also going to get an increase in water movement across aquaporins, ADH, right? In the DCT and collecting duct, the water permeability is dependent upon aquaporin 2s, and aquaporin 2s are dependent upon ADH. So we're going to get, I'm not going to squeeze those in here, we're going to get water movement as well, increase in water movement into the extracellular fluid. Okay, so we just boosted our blood volume, by boosting blood volume, we're boosting our blood pressure through increased central venous pressure, through increased venous return, through increased cardiac output, things we've been through before, right? And so there's where our tie-in to solving the hyponatremia and helping with our low blood pressure. Now, we have to solve this hyperkalemia bit. Well, we had the potassium going around and around. Well, one of the other channels that can get expressed, especially with higher aldosterone secretion, is a channel called RAAS, and that stands for renal outer medullary potassium channel. Now, I didn't list that here because it's not a major target until the aldosterone levels get really high. But what happens then is some of this potassium, the high potassium inside the cell, will actually travel this way as well. Right? And there are always potassium channels there. There's just, we're adding even more of them when aldosterone, the stimulus for aldosterone is especially high, when we have significant hyperkalemia. Then we really enhance that swapping of sodium reabsorption for potassium secretion. And that's really kind of one of the key things you want to think of aldosterone doing. It's retaining sodium, and in doing so, it's setting us up to secrete potassium. And so it's solving the hyponatremia, it's solving the hyperkalemia, because sodium is the primary extracellular cation. Retaining the sodium in the ECF helps us retain water into the ECF, so we get a boosting of our blood volume, which boosts our blood pressure. All right? So again, primary actions here are probably electrolyte balance, mostly potassium, but the retention of sodium in response to aldosterone significantly helps with the blood volume elevation and tending to boost blood pressure. Remember, ADH is off doing its thing, right? Down at the medullary collecting duct down below. It does it up in this region too, but it's more effective down in the medullary collecting duct because that's where that interstitial gradient is, right? Remember, that's why we have a loop of Henle. So the ADH is even more effective down there, and that's not dependent upon sodium movement, right? That water movement. That's just adding channels to enhance the permeability there to boost our blood volume. So we're really boosting blood volume in two different ways, with aldosterone via the sodium retention and ADH by just increasing water permeability without enhancing ion reabsorption. Okay, I think we're good with the renin, angiotensin, aldosterone / ADH system.